Uh, I'm really happy to be here. And before just taking this warning away, before the first thing I want to say is that I started feeling under the weather during lunch. That's why I'm like with a cupboard on my back. But I'm really excited to be here, and it's really nice. I feel I have a special connection with Bonn because my PhD advisor was there many years, Torsten Struck, before he moved to Oslo. I think it was his final location in Germany before he reallocated to Oslo. And now a master student of mine who finished 3 years ago, Nina, she's a PhD student with you guys. So I'm very excited to be on Zoom, but I will be very excited to visit you guys, especially because, there are so many familiar faces here. Today, I will be talking about some of the work I have been developing and the things that I have been thinking for the next few years, mostly about ecological niche dynamics, adaptive radiations on oceanic islands. And the first thing I want to start really at the basic level, so we're all on the same page. If you're unfamiliar with this concept of adaptive radiation, it's just this a single species, usually a common ancestor, usually a generalist, as the case of this finch here, um, colonizes a new area or has access to new ecological resources. And then there's like this chain reaction of speciation and ecological niche specialization happening. So in the case of the finches, you have the evolution of insect eaters, seed eaters, cactus, seeds, plants, and buds and fruits. And so adaptive radiation is a bit where speciation and ecological change, ecological specialization is actually amplified multiple times. And that is why a lot of the classic systems have given us so much, but so much of what we know about the diversity of life. And so I wanted to highlight some island examples, but there are the cichlids, there are so many other adaptive radiations, the Loxia crossbills, etc. And they give us a lot of insights about biology, mostly because speciation and ecological things are happening. And then we can look at the things happening and make hypotheses. Why is this before that? Why is this happening? Is this leading to that, etc.? So in this case here, just to comment the images, you have the same as the Darwin finches, where the common ancestor for the Hawaiian honey creepers just radiated through the whole of Hawaii. And then you have the evolution of different beaks. Um, in this case, they also do nectar feeding and all of that. It's a very beautiful radiation. And this is a classical system. If you don't know it, it's the anoles that they exist throughout the whole of the Americas. But in the islands of the West Indies, so the Caribbean islands, Puerto Rico, Jamaica, etc., they have repeatedly evolved these ecomorphs. So there's usually like one species that comes and colonizes and then evolves rapidly or not, that's a bit disputed in the knolls, but evolves these four forms that then are segregated in the oh no, I have the reactions on that. That segregate in the ecological niche. Adaptive radiations tend to occur on islands, and this is why we focus on islands. So this was a paper we published last year, and where we have like these beautiful archipelagos. I like to say that this is kind of my holiday map, the places I want to go on holiday. And so you see that the number to the left is the number of papers studying an adaptive radiation. And the second one is the number of adaptive radiations. And what you take from here is that, first there are many, but of course the places that are associated with countries where there's more research, such as, for example, Spain, the US here with Hawaii, there will be more adaptive radiations found because more people are working there. Classical places like the Galapagos will also have this. So adaptive radiations do occur a lot on islands, and this is because of the special conditions of islands, which I will be talking a bit about in a second. Because there are two cool things about islands, I would argue. One of them is the adaptive radiation, and the other one is what we call the island syndrome. And these things are not mutually exclusive. So the island syndrome is just this idea, and this is a very simplified, I have it very simplified here on the slide, but that lineages on islands become phenotypically very distinct. And these are things that we observe repeatedly. So, for example, birds repeatedly lose the capacity to flight. Larger mammals become smaller on islands. So, for example, we have pygmy mammoths evolving convergently or in parallel, depending on what terms they use repeatedly. And so I like showing this image to my students so they can gain an appreciation to how alien and scary, I guess, things can look like. Like the cursed lemur, yai. I just learned about this in Jonathan Los's book, Improbable Destinies, and the coconut crab, and then these cool plants like the one to the left is an opuntia in the Galapagos. That's one of the few trails you can do in the Galapagos, and they have these really beautiful cacti trees there. And then the one on the right is the Socotra dragon blood trees, the one on the back, the taller one, and the one on the front with the pink, this guy here is the elephant leg tree. So things just look weird, island syndrome, and things just like to speciate and become distinct, adaptive radiation. And these are not mutually exclusive. And so going back, this happens because of the particular conditions of the islands. Let me enumerate. There are discrete habitats, variation habitats, isolation, disharmonic biota, and empty ecological niches. So let's look at this. This is the big island of Hawaii. So Hawaii has several islands. It's an archipelago, then it has the island of Hawaii. And you see that I can draw a line from the side that gets the trade winds and where you find humid forests, and then the side where the air has no humidity. And so I wouldn't call it a desert, but it's a very arid landscape. And so you see here how discrete these habitats can be, how extremely variant the habitats can be. But then when we look at the Galapagos here, and this is the island of Isabela, Fernandina, Santa Cruz, and Pinta, sorry, you see that we have these pockets of the green here, which are the humid highlands. So these environments occur repeatedly on the islands, and they're associated with the trade winds. Here they are on the south because that part is on the south of the equator, and then this on the north because it's on the north of the equator, which is quite cool about the Galapagos. Then there's the isolation, which leads to a disharmonic biota. So there are lineages that you do not expect to find on islands, and this is what we call the disharmonic biota. So, for example, some of the first colonists are spiders, but then you don't expect to find amphibia, and the only mammals that you expect to find are bats and humans. And um, this is because of the ability to survive long distances. Spiders have they fly with their silk, but amphibia, if they touch salt water, they die an egg. So they don't manage to colonize these remote and isolated islands. And this leads to the fact that there are empty ecological niches. So, for example, there's this island that has the largest insect in the world, and what has been speculated or shown or commented upon is that it's filling the niche of a rodent. It behaves kind of like a rodent, it does the same things as a rodent, and there are no rodents there because rodents were not able to colonize. So then that niche was open, and it was free real estate, so to say, for an insect. And so just to step a bit back, and so we're all on the same page before I give you a bit more, is that the ecological niches is the space occupied by species in nature, and an adaptive radiation just is this occupation of progressive ecological space. So in this example here, we have three warblers, and you see this warbler is occurring here on the middle of the tree. This warbler is near the tips on near the top of the tree, and this warbler here feeds on the lower part of the tree. So they occupy different parts of the niche, and this is how they chose, so to say. This is how they partition their niche. And so if we go back to island examples and we put this on a graphic, like this very cheesy graphic that I made with a dinosaur, we have environmental variable 2, environmental variable 1. So if you like to think about plants like I do, we can think about this being, for example, temperature, and this one being precipitation. And so high precipitation and lower temperatures, and so forth. You have a colonizing species, and the colonizing species expands in population size. Why? Mostly because you're coming to a new place. There are not a lot of big predators. Sorry, there are not a lot of predators or enemies where you just have arrived, because part of the island syndrome predicts that species are not going to compete as much because there's more ecological space there open. And so there's this expansion of the population size. And then you might have these generalists, and it's usually generalists that we presume that start radiations. But then with the increase of population size, you're going to have more genetic variation being generated, and then there is a higher likelihood of evolving new phenotypic combinations, and that some of these are specialists. And these specialists perhaps the specialist here, in this case, I have camouflaging. It might be do better and have a higher fitness, and then it might expand population size, and then this might cycle again. So then we can have this progressive occupation of ecological environments. One thing that is really well established in adaptive radiation is that there's a correlation between ecology and phenotype. So here we have the perch size and the limb length. So the limb length is just morphology of how long the limbs are, and the perch size is where they are on the tree, like a bit like the warbler example that I showed you. And so you have three specialist species that have adapted now to the island, but let's say that these don't exist. If there's a new generalist colonizing, part of the population might have selection for shorter limbs, and part selection for longer. And so we have this correlation between ecology, specialization in a niche, and the morphological changes. So then, after giving you this, a brief walkthrough, one thing that I noticed and that I got interested is about the sequence of niche occupation. Is it random how, if we go back to this example, would the yellow evolve before the green or the green before the yellow? If we ran a simulation, would it be random, or is there a higher likelihood that the yellow evolves before the green? Is it because perhaps evolving on this niche axis is, for example, could be easier, so both of them would be easy, and then on this axis is hard? And I was thinking a bit about these ideas. And so I made this thought experiment that I wanted to walk you guys through. Imagine that these ellipses are different niches. And again, if you think about plants, sorry for the zoologists, I like to think about the climatic niche because it's simple. So climate, temperature, precipitation. And we have this continuum of extremely arid, very arid, arid, humid, very humid, extremely humid. And now let's presume a pattern that is random. So we have this colonist from the environment. It's a generalist, but it's on the continent. It occupies more arid regions, so it establishes original population in an arid part. And so in a random thing, this could be random, right? So for example, it could jump to very humid. The one from very humid could just jump and occupy extremely humid and very arid. So I'm assuming that speciation events occur, at least some degree of specialization, and then some degree of population structuring and reproductive isolation, perhaps. Um, and then this guy with the very arid would go to extremely and to humid. So this would be a case for the random. However, we can also have the case where you have a progressive one, right? You have this colonist from the continent that then goes like this and occupies the ones nearby, and like this eventually. And so the question that I have now for the 10 minutes more that I'm going to be talking about this is whether is ecological occupation progressive or random? And so I was thinking a lot about how to test this, and the way that I came to test about this, but I'm open to ideas and to refining analysis and all of that and collaborating, is that I'm interested in branch lengths, so phylogeny, the divergence of the species, and ecological data. And it has to be quantitative. So for example, the branch length between, this is a clavicle, so it's not the best, but it's just a dummy example. So in this case, you have the branch lengths would be this and this, between 1 and 2. Between 1 and 3 would be this and this. And so we just get pairwise branch lengths, and then we would get pairwise quantitative measurements of ecological divergence, which here you see hot habitat, intermediate habitats, and cold. But for this, I'm showing you again a very simplified example, but this will have to be quantitative so we are able to make correlations. And the expectations I have for this question is if we have a progressive model, we would have this, whereas a random model, we would have something more like this. Okay, that has been 14 minutes to get to the to the answer. And so for Darwin finches, the carbon 13, which I think, I mean, I did this very quickly, but I think this is a very important measure of what they eat. We have a correlation of 37%. Not every variable comes out. I think this variable is not so associated with diet, so we don't have this thing. So I just use this kind as a control, so to say. Um, and one thing that I actually did was that I went to the genera inside the finches just to see, because the finches is the radiation where we have most data from. And so I was interested in looking at this intra-genera analysis. So this one is, I don't remember the genus, sorry, but this genus here, and then this genus here, I took the species like this, and then I just looked whether, at this closely related species, whether we were not observing some, things that happen after speciation, whether things are happening as we look. So I wanted to look closer in time. And we have the same type of pattern that we observe. When I do the same with anoles, we have the same pattern. So again, the perch height and the phylogenetic distance, when I consider the adaptive radiations separately, because anoles is a very large genus. I think it has 200 or 300 reptiles in North America, South America. But on the adaptive radiations, it's following this pattern. On the Canary Islands, what we observe is with this climate data that was pre-processed by a paper and collaborators, this IOMune, they also have this, let's call it beautiful correlation. And then on the Scuriania in the Canary Islands, we also have this correlation. And the one before was the climate data. This one is elevation, but we also have with precipitation, temperature. I just don't want to, actually, I have one more graphic, but I don't want to bother you because this is precipitation on the cold quarter, which is, yeah, slightly more inclined. So then I got very interested in what are the mechanisms of the occupation of ecological space and what mechanisms underlie this. And so one hypothesis for this is geography. So it's easier to transition to closer niches because they are geographically closer. So if we look at the map of the Galapagos, you see that the humid highlands here have a transition zone, right? And that so it might be easier to jump there and then there rather than to jump there directly. And this is verified even if you say, but you say in San Cristobal and Santa Cruz, yeah, but those islands are older. There has been a transition zone there, but the island, as they age, they start eroding and they start becoming smaller and smaller, so they start losing ecological areas because the islands start sinking because they leave the hotspot and they start eroding. And here you have like a representation as well that you have like the geographical proximity between these different qualitative niches and and what we observe with geography. Another mechanism that could underlie this is that it could be organismal constraints. So phenotypic constraints, lack of genetic variation, genotype, phenotype might being complicated, it might be easier just to change a tiny bit than to make very big leaps. So it might be easier for Magni rostus to evolve to become like Fortis, Fortis to evolve like Perdula, and Perdula eventually like Certhiadea, here Oliviaceae, rather than this guy jumping all the way here and then like this and then like this. So these are hypotheses and I have contacted a theoretician who got very interested in this, and we're going to be doing some models about this. Okay, that was my part one, and sorry for having a bit disconnected. I would say they're quite connected, but I'm just shifting gears a bit. The part two that I have, I wanted to talk a bit about genetic diversity and genomic structures that might be responsible for the variation that we observe. So I briefly alluded to this that, like we have these alien things, we have these adaptive radiations, we have these beautiful landscapes, and the evolution of these extreme phenotypes, right? And one thing I would like you to appreciate, and maybe it's the first thing you think about, but it might not be so obvious as well, is that islands are small, isolated, and remote. So you see on this image here, Hawaii is like 4,000 kilometers from LA and 5,000 kilometers from Tokyo. So it's hard to get there, and the areas are small, right? And so we know that the populations that live there should also, in theory, be small and geographically restricted. And there are many papers showing this, calculating NE and all of this. And if you work with population genomics, like I do, some of the central tenets that I guess we need to know is that natural selection is inefficient in small populations because there will be a lot of fluctuations and selection will have a harder time to counterbalance the effects of genetic drift. And with this, you also have a higher accumulation of deleterious mutations because selection is not there, is not efficiently removing this, right? So both the effects of purifying selection, cleaning all of this, and the effects of positive selection diminished. And this is what I call the island paradox. And I was very lucky to get the project funded on this earlier this year. So there will be more coming about this. I'm trying to be invited next year as well because it's so nice to give departmental seminars. So the island paradox is just this idea that how can some of the most diverse lineages exist on isolated areas with small populations, and how does this work? What is natural selection picking on? Like what is changing? And for example, a lot of the classical adaptive radiations, like the cichlids, we know that hybridization has occurred. Some stuff comes from this river. Some stuff comes from this river. It becomes a slot machine with a lot of things happening. Really brilliant radiations, but these are more isolated, I would argue. At least something like Hawaii or something like the Galapagos. And so a more testable or scientific question to the island paradox is what mechanisms generate and underlie this genetic diversity on islands. Last year, and I alluded to this paper before, but last year we published this paper where we have some textbook stuff. This is nothing new. The mechanisms that create genetic variation are de novo mutation, hybridization, sorting of polymorphisms, chromosomal rearrangements, polyploidization. Just FYI, for your information, I think there's an island in the Pacific. It might be the Juan Fernandez or Hawaii. I forgot. I have a horrible memory. That 70% of the plants there are polyploid. So there seems to be some advantage in polyploidization, or polyploids seem to have some advantage in colonizing. Um, and transposable element insertions. And the one that I will be focusing a bit today about is this one about transposable elements. Again, so we're all on the same page. If you don't work with genomics, transposable elements are these things, these jumping sequences. So if you have this red sequence here, and then you see this blue one with a turquoise middle, the turquoise part jumps from the blue to the red, and you go from this scenario on the left to this scenario on the right. Sorry, I have a red surface here. And so you just have like the reallocation of genetic variation. And the way that I like to explain this concept to my students is that these are genomic viruses. They are copy pasting and cutting and pasting and rearranging things a bit in a selfish way um, to the organism and trying to perpetuate themselves. And so transposable elements can generate genetic diversity by changing the genomic makeup. And this would be what some people would call large mutations. So for example, changes in promoter sequences, frame shifts, intronic modifications, gene duplications. So for example, if they jump in front of promoter or in the promoter, they might just deregulate it or change it. Frame shifts if they jump inside a gene, if they jump inside an intron. Sorry, this one would be an exon to separate from this one, intron. And then gene duplications, which sometimes T's just jump and then recombination kind of messes up and then duplicates that region because it misaligns, because it's like, oh, this T is here, this T is here. Let me copy everything in the middle. And it's actually a quite common way that genes become duplicated other than polyploidization. And if you're not convinced, or if you don't believe me, the iconic case of adaptation or textbook example is the result of a transposable element that jumps into a gene and that causes, I don't remember if it's a frame shift, but causes modifications in the expression, and then eventually the melanic morph, or not eventually, the melanic morph evolves. However, there's always a but. However, when I was interested in T's, I got a bit frustrated because a lot of the literature is what I call macro, in this way that is not very critical, I would say. So people are just like comparing species-rich lineages and saying things like species-rich lineages have more T's, speciation occurs because of T's, adaptive radiation occurs because of T's, and a lot of this works. So just to give you examples, that they're comparing mammals versus marsupials, or even like platypuses, passerines versus a very less diverged bird lineage. And then, they get like these significant correlations, and they're like, T's must be driving all this diversity. It must be T's. But there are two things. First, correlation is not causation. And then second, um, they have so little genomes. A lot of these papers are still being published, but a lot of these papers are like, we have four mammal genomes and four, and like a platypus genome. The mammal genomes have much more transposable elements. Therefore, transposable elements are what made us not platypuses and made us radiate throughout the Earth. And so I got interested in this question of, can transposable elements help us understand evolution on oceanic islands? And the way that I approach this, and I'm repeating some part of this, is that we use this Illumina low coverage data plus a repeat library from a database or a nearby genome. And what we have been doing is, so to say, very simple. DNA pipe TE says, transposable elements should be highly repeated on the genome, right? So when you give it the baseline of 0.5x, it just says a lot of the stuff that is represented less than like that is not so much represented is not going to be a transposable element. So it fetches those reads, it assembles them, and then it identifies them. And it has been quite good in identifying them, as I will be showing you in a couple of slides. But before that, I'm going to give you just a brief introduction to the system. This is part of the systems that I'm focusing on the Galapagos. We're now expanding to new systems as well. But the Galapagos Scalesia is a very diverse lineage, has 15 species from tree shrubs that evolved from a common ancestor lineage. And, like in less than 1 million years, in plants, we usually love looking at leaf. So this would be like the, free jaw. Sorry, I don't know how to say the jaw of the cichlid. So this would be like the variation that we observe on jaws of cichlids and other things and on anoles in terms of limb length. But you see that all of this variation is evolving, and this is variation that usually doesn't evolve so quickly, and this variation that some papers have established now that is correlated with climate. One thing, and this is a paper from my collaborators, one thing I wanted to appreciate is if you look at H and I. These are not sister species, but very closely related species. This is a forest of Scalesia pedunculata, which is, these are trees that are 12-meter tall. And this is, I believe, a Scalesia finis in Isabella. And so these guys are occupying this, impenetrable or quasi impenetrable lava flows where cacti live, or you only find cacti in Scalesia, and also making their own forests. And, I can guarantee to you that you can drive for 10 minutes and see both species, which is crazy to me that you think that these closely related species are so close also geographically, but they're so morphologically distinct. And one thing that we were particularly interested in exploiting is this ecological dimensions of the climate that I have alluded to several times. So in this phylogeny here from our collaborators, Mario Masuecos et al., Masuecos Fernandez et al., they established that Chordata and microcephala are present in the humid highlands. So the blue one is dry and low lands. So very dry, close to the coast. The red and the orange means that they have colonized the humid highlands, and there are some also on the dry highlands. So what I wanted to retain is that there has been at least two adaptation events, presumably, or there have been two adaptation events to being able to go up and do these forests and like adapt to this uh, completely distinct ecological conditions. And in the context of the ecological climate, and now I will be talking a bit about the hypothesis we have, is that if you live on a humid highland or a coastal region and you're a plant, you will be subjected to remarkably distinct temperature and sunlight regimes. So, for example, if you're one of these calicias like a finis next to the cacti, you're going to endure 40 Celsius all year. The lava flows are not going to give you a lot of water, and they're not going to open up for you to take a lot of nutrients and things, while the humid highlands has this permanent moisty, foggy conditions that are humid sometimes the whole year. And so I would argue that there's a very big selective pressure on retaining water between these two ecological niches, which is what we expect for plants. And when we publish the genome, the cool thing about this paper is that we separated the polyploid genomes, the subgenome A from subgenome B, But we found out that when we ran these tests of genomic expansions, so expansions of gene families, we find a lot of expansions, so a lot of novel genes or duplications of genes associated with aquaporins, with water retention, etc. So there should be a very big selective pressure for water. And we reasoned transposable element accumulation is going to lead to a larger genome size. Larger genomes are one-to-one correlated with cell size and with stomata size. When you have more transposable elements, you're going to have more DNA. More DNA means larger nuclei. Larger nuclei means larger cells. There's a one-to-one correlation in plant biology associated with this. And if you have larger cells, you are worse at water economy because you need to get more water for the to sustain yourself. And if and the other drawback, so to say, is that your stomata, where you make the changes with the environment, with the water vapor and etc., are going to be less efficient because they're going to be fatter, so they're not going to be as efficient. So we hypothesized one that there should be differential accumulation of Ts in lineages adapted to different ecological niches, and then we hypothesized two other things. One of them I haven't alluded to, and it's on this slide. It's about hybridization. When Barbara McClintock published her seminal paper, she put that, perhaps hybridization, and I think drawing from Meyer and the ideas that hybridization destabilizes the genome, she put out saying that hybridization should lead to uh, disestabilization of the genome by release of transposable elements. And we know from Scalesia, and sorry, I haven't mentioned this before, but we know from Scalesia that we have hybrid populations that we can test this. So we looked at ecological niches and had hybrid populations. And the third thing we were interested in is just the comparison between the outgroup and Scalesia. The outgroup has been suggested as a fast radiation as well, but Scalesia should be much younger. So if transposable elements are associated with the radiation, we should find many more transposable elements in Scalesia in comparison to the outgroup. So we have these three hypotheses. So then here, tree first. These are the outgroups, palpable bolus, and this is Scalesia. These are the hybrid populations. The hybrid populations have roughly the same T content. In different colors, you find different T classes, so DNA, electrons, LTRs, and nomes, which are important, I guess, to mention, are this here, with Scalesia villosa. And so the hybrid populations don't have more. And then the species present on the humid highlands, which would be pedunculata here, doesn't seem to have more or less. And the other one associated with the humid highlands, which would be these guys here. Damn it, sorry, I have such a rugged mouth surface. Basically, I would argue they're all the same. I didn't make any p-value statistic, but I would argue that they're all the same, which doesn't really go in, which just kind of refutes every hypothesis. And then when we look at the species, at the accumulation through time, so on the x-axis here, we have the cumulative substitution level. If you are used to this type of data, you know that this is just a very indirect measure or indirect measure of how divergent they are. So this would be a measurement of genomic divergence, and then you have here proportion of the genome. And you see that the outgroup in green is different from Scalesia. The species in Scalesia barely no changes, with the exception of this one that I really don't know what might be happening there. Then the thing that I did afterwards was that I looked at groupings of transposable elements in a different way. So I got all transposable elements from all Scalesias and I grouped them in orthogroups, so in groups of orthologs, so to say. And then I put them all together and I made this Venn diagram. So this is a Venn diagram on steroids. And this Venn diagram, this subset plot, what it shows is that the first category here is, I'm trying a new surface here, but T's groupings present in all species, 4,387. And then you have this huge leap to species that are single. So for example, the second group is Scalesia retroflexa helleri hybrid that has T's that were only present there, only 195, and then this one until we get to 142 in Croceri aspera hybrid. And then these categories here are just going to be present in all, but the first one, which is Crockeria and Haspera. This one would be present in all, but I think this is Gordiloi if I'm doing the horizontal as well. So a lot of the Ts seem to be shared, and there doesn't seem to be like any private family really going loco on the genomes. When we look at the quantity of Ts, there's no difference at all. So here we have again on the x-axis the species, and on the y-axis, I have to convert this to a table. And on the y-axis, we have counts. And so the even colors that you find horizontally means that there is no particular enrichment of transposable elements uh, on any species. So, for example, LTRs have the same count, which, judging from this color, is something around 5,000. Um, LTRs that have not been identified, it's around 5,000. But then copias and gypsies are on this, but with very little variation. So even if these are very high numbers, we find little variation. So the conclusion from this part is that different ecological niches do not seem to promote expansion of constraints of TEs. We don't find this correlation ourselves. The hybrid lineages do not have more TEs, and the outgroup does not have more TEs than Scalesia. And so maybe a provocative thing to say is that we should be careful with these macroevolutionary and macro ecological suggestions when we have so big correlations and so little data. And my last slide, I've been talking for 36 minutes, is that as Madeline said, I'm going to Sweden. So if you are interested by what I presented or what I do, don't be a stranger, reach out. There's a PhD position in Diplotaxis in Cabo Verde. It's plants close to Arabidopsis. Currently working on getting the permits and getting the student running. We have 300 plants in Oslo ready to go. One PhD position in Birds of Paradise, if you're interested in that, with colleagues in Stockholm, and then two postdocs in theory of adaptive radiations, hybridization dynamics. So if you like doing individual-based models and stuff, and theory, please reach out and I can talk to you. The first two should be announced around January, and then the last two, so the postdocs should be announced around March or April, because we were afraid of the administrative load of four positions. So thank you for your attention. And this is a beautiful Scalesia, I think, affinis, with its not flashy flower, because flowers don't tend to be flashy on islands. Things are weird there. So thank you.